Okay, so let's talk about the RNA synthesis or the transcription. We are primarily describing the RNA synthesis or the transcription as occurs in prokaryotes. Some important differences we'll list after we have completed the whole process, as we see in the eukaryotes. We'll display some of the important differences as we see in the eukaryotes. Okay, so first let us discuss the transcription as occurring in the prokaryotes, and then we move on to the eukaryotes. Now when we are describing the transcription, we discuss the transcription in various stages. First we have the initiation. First we have the process of initiation. Initiation is followed by elongation, and finally we have what is known as the termination. All right, so we have the initiation, elongation, and the termination. The three mechanisms are initiation, elongation, and the termination. Let's start with the process of initiation. Now the transcription will be carried out by the RNA polymerase, obviously. And in prokaryotes we have only one single type of RNA polymerase. In eukaryotes we had seen we have the RNA polymerase 1, 2 and 3, but in prokaryotes we have one single type of RNA polymerase. This RNA polymerase is made up of the 5 subunits: 2 copies of alpha, 1 copy of beta, 1 copy of beta dash, and the omega. This is the active RNA polymerase. It is ready to transcribe. It is ready to transcribe, but the problem is it doesn't know from where to start the transcription. So just like all of us are getting so restless, now we want to go out, we want to do some activity, maybe you want to do a party, but you don't know where the party is. Everywhere, because of this COVID crisis, nobody is holding the parties. Okay, so it is very difficult to find out where the party is there. Same way, the RNA polymerase is ready to transcribe, but it doesn't know from where to carry out or start the initiation point. It doesn't know from where to initiate the transcription. All right, so RNA polymerase alone is not enough. We bring in another factor, and that is the sigma subunit. We bring in the sigma subunit. When we add the RNA polymerase to the sigma subunit, what we get is known as the RNA polymerase holoenzyme. Alright, so now we get the RNA polymerase holoenzyme. So the RNA polymerase, the functional enzyme, plus the sigma subunit will create the RNA polymerase holoenzyme, which also has the information from where to start the transcription, because the sigma subunit is able to identify the promoters. One important thing to note is the sigma subunit is specific for one promoter, meaning if you have, let's say, 20 genes or 30 genes or 40 genes in the prokaryotic organism, then you are going to have as many sigma subunits, because one sigma subunit is able to identify one single promoter. For every promoter we will have a different sigma subunit. Whichever sigma subunit gets attached to the RNA polymerase, the corresponding gene is going to get transcribed. Alright, so sigma subunit provides the specificity for the promoter in the prokaryote. This question is asked by the examiners: how which unit will describe, or it will provide the specificity for the transcription. So your answer should be the sigma subunit. Okay, so now we have the RNA polymerase holoenzyme. Let's see what happens now. We are talking about initiation. So we have the DNA like this. We have this DNA like this. Now RNA polymerase holoenzyme will scan the DNA. It will scan the DNA and it will look for the corresponding promoter site which matches, or for which the sigma subunit is specific. Once it is able to locate that particular promoter, then it will go and attach to that promoter site. Let's say this is the sigma subunit, this is the sigma subunit. So the sigma subunit will identify the corresponding promoter will identify the corresponding promoter, and the attachment will be there between the sigma subunit and the promoter. See, the rest of the RNA polymerase is not attached to the DNA. Only attachment occurs between the sigma subunit and the promoter. Once this attachment occurs, the rest of the RNA polymerase will initiate the function of separating the two strands. First thing which happens is the two strands of DNA will undergo denaturation. So now a segment of DNA is denatured. Now look at this structure. This very important structure is known as the pre-initiation complex. It is known as the pre-initiation complex of transcription. As you can see, two important points to note. First, the RNA polymerase attached to the DNA and the DNA is denatured. So the pre-initiation complex of transcription is the RNA polymerase attached to the denatured DNA. You can see the DNA is denatured here. You can see the DNA is denatured here, and the RNA polymerase complex is attached. Alright, so pre-initiation complex of transcription is formed, and now the RNA polymerase will transcribe this small segment. It will transcribe this small segment. The size of this segment is about 8 to 10 nucleotide long. The size of this segment is about 8 to 10 nucleotide long. What is happening? This RNA is attached to the DNA and the RNA polymerase is attached to the RNA. So previously the RNA polymerase was attached only to the promoter via the sigma subunit, but now the RNA polymerase is also attached to the RNA which it has synthesized. Now using this RNA which has been synthesized, what can happen now? The RNA polymerase can start moving forwards. Imagine first it is holding this particular RNA, then from here it jumps to next RNA nucleotide, then to the next nucleotide, then to next nucleotide, and then to next nucleotide. What it means is now the forward movement of the RNA polymerase is possible. It gets the scaffold. Previously it did not identify anything, it was identifying only the promoter, so it was fixed. But now when the RNA synthesis has started, the RNA polymerase is able to move forward. When it is able to move forward, we call it the promoter clearance. We call it the promoter clearance. So promoter clearance is the last step of initiation when the RNA polymerase will start moving forward along the DNA. It will start moving forward along the DNA. So what will happen? It will move forward. Let's say after some time you will find what is happening. Now we are entering the domain of elongation. So what will happen here? The RNA polymerase after some time will be like this. It has moved along the DNA, and when it goes forward it will cause the denaturation of the different segment of the DNA, and as it keeps moving forward, it keeps carrying on the process of transcription, so that our RNA is becoming longer and longer and longer. You can see our RNA is becoming longer and longer and longer. This is the process of elongation. Where the RNA polymerase continues to move forward. It causes continuous denaturation of the DNA, and it keeps adding the ribonucleotides so that the RNA is extended. This is the process of elongation. Nothing much to describe elongation. Only thing is, after promoter clearance, the RNA polymerase is regularly moving forward, and it keeps causing the denaturation of the DNA and extending new ribonucleotides. All right. So this is the elongation. Now the termination can occur in two ways. The elongation will continue until one of the two conditions required for termination is fulfilled. Termination can occur in one of the two possible ways. First is rho or ATP dependent termination. The first one is rho or ATP dependent termination, and second one is obviously rho or ATP independent termination. Alright, so for both of the termination process, special conditions have to be fulfilled before the termination can occur. Let's see what happens. First, let's talk about the rho dependent termination. What is happening in the rho dependent termination? There is a transcription of a rho binding site. A segment of DNA has been transcribed into the RNA, where a important subunit called the rho can come and get attached. So here the rho subunit will come and get attached. This is the rho subunit, right? So the rho subunit will come and get attached, right? This rho subunit has the special property, it is able to cleave the hydrogen bonds, that is the interlinking between the RNA and the DNA. It is able to cleave the interlinking between RNA and DNA, for which ATP is required, that's why it's called the ATP dependent. Rho factor is required and ATP is required, so it is the rho dependent or the ATP dependent termination. The special condition is rho binding site is transcribed. When we transcribe the rho binding site, the rho subunit will come and get attached, and with the help of ATP it will separate the RNA. So let's try to summarize what is happening. In case of rho or ATP dependent termination, the first condition is the rho binding site is transcribed. When the rho binding site is transcribed, the RNA cleaved from DNA using ATP. Who's doing the cleavage? By the rho subunit. The rho subunit will cleave, it will cleave the RNA from DNA using ATP. Now, very important point to remember that our RNA polymerase was attached to the RNA, it was not attached to the DNA. On DNA, it has only one binding site, and that is the promoter site. But we already moved away from the promoter, so now the RNA polymerase is attached to the RNA. So what happens? RNA polymerase, which is also attached to RNA, or rather you can say which is attached to the RNA, will obviously also separate. It will obviously also separate, meaning now we are looking at termination. Once the RNA polymerase will separate from the RNA, that is the termination. So the rho binding site will separate the RNA. RNA is attached to the RNA polymerase, so the RNA polymerase will also come out, and that is what is termination. Then the whole process can start again. The RNA polymerase will attach with another sigma subunit, and again the whole process of initiation, elongation, and termination can occur. Alright. So this is one of the conditions, that is the rho or ATP dependent termination. This is the rho or ATP dependent termination. All right, so now we go to the second category, that is a rho or ATP independent termination. The condition for the rho or ATP independent termination is slightly different. Obviously, it is slightly different, and now we are talking about the rho or ATP independent termination. Here, two very critical conditions have to be fulfilled. first the first condition to be fulfilled is we must be able to replicate a GC rich segment which has a specific property. It is self complementary. So GC rich segment which is self complementary, what will happen if you transcribe? See, these are two segments which are self complementary. So once their transcription is complete, they will start pairing up. They will pair up like this because they are self complementary. So they will pair up like this. All right. So once they pair up, they will start pulling on the surrounding tissues. So this GC rich segment is immediately followed by a weak segment, means it is a poly U segment. So two conditions: first the GC rich segment, and then the poly U segment. So you have the transcription of GC rich segment, which is immediately followed by the poly U segment. If these two conditions are fulfilled without the need of any energy, immediately the termination will occur. So you can see it is the rho independent or the ATP independent termination. It is the rho independent or the ATP independent termination. So we are talking about the second, that is the rho or ATP independent termination. This will occur when two conditions are fulfilled during transcription. What are those two conditions? First is the transcription of a GC rich segment. All right, transcription of GC rich segment which is self complementary. I missed that. Transcription of the self complementary GC rich segment. All right, transcription of the self complementary GC rich segment, which is immediately followed by the poly U segment. So like I told you, what happens? So these are the GC rich segment which are self complementary. They will pair up and they will start pulling the rest of the DNA. They will start pulling the rest of the DNA. The poly U segment will be like the AT rich region that was there in the DNA. It is weak because only two hydrogen bond between the pairs will be there. So it is going to separate on its own without the need of enzyme. It will separate. So once the RNA will separate, the same thing will happen, and that the RNA polymerase will also come out because it is attached to the RNA. All right. So here what is happening? The ATP and rho independent termination is happening. ATP or rho independent termination is happening. So the whole process is very similar in the eukaryotes with one very important difference. There are no sigma subunits in eukaryotes. There are no sigma subunits in eukaryotes. So what do we have there? We have the transcription factors. Now these are big complexes. The transcription factors are big complexes and they are multiple in number. They can be numbered and they can be having, each of them can have the subtypes like this. All right. So we have the transcription factors which can be numbered and have subtypes. They are very complex structures which will regulate the transcription in the eukaryotes. In the prokaryotes, the regulation was by sigma subunit, but in eukaryotes, the regulation is by the transcription factors. Out of these transcription factors, one of the very important transcription factors is what is known as the transcription factor 2D. The transcription factor 2D, which itself is made up of the two components known as the TBP and the TAF. The TBP and TAF. The TBP stands for the Tata box binding protein. It stands for the Tata box binding protein. In the promoter segment, you know that there is a sequence called TATAAG, which is known as a Tata box. So this protein will go and attach to the Tata box in the promoter. In this way, the promoter can be identified. All right, and then we have a whole host of molecules which will come when the TBP gets attached, which are known as the TBP associated factors. All right, so together the TBP and TF will make up the transcription factor 2D. So they will make the transcription factor 2D. We already said in the eukaryotes we have the RNA polymerase 1, 2 and 3 rather than a single RNA polymerase, and we had seen when we are talking about RNA that each of them is responsible for the transcription of a different type of RNA. They are responsible for transcription of a different type of RNA. So at this point, let us talk about some inhibitory molecules. Sometimes you can get this question about the inhibitory molecules in transcription. Inhibition of transcription. Alright, this is sometimes asked as a 2 or 2.5 mark question, and describe the inhibition of transcription maybe in the prokaryotes. Primarily it is asked for the prokaryotes. So here you have to remember the inhibition of transcription can occur in two ways. One, some of the inhibitors bind to the beta subunit. You recall in prokaryotes the RNA polymerase have 5 subunits: alpha, 2 alpha, 1 beta dash and omega. So some of the inhibitors bind to the beta subunits. The molecules include, obviously you will read about these drugs in detail next year, I'm just giving you the name: rifampicin, the rifamycin, the streptovaricin, the streptoglycidin, and even heparin. All of these will bind to the beta subunit of RNA polymerase. They bind to the beta subunit of the RNA polymerase. Then there are some molecules which will attach to the DNA and prevent the attachment of the RNA itself. So there are some molecules which will attach to the DNA template. They will attach to the DNA template and prevent the attachment of the RNA polymerase. The one important example that we have here is known as actinomycin D. Sometimes it is also known as the dactinomycin. So both the names are used, the actinomycin D or the dactinomycin. Alright, remember. So this is the inhibition of transcription that is seen in the prokaryotes. For eukaryotes, we have already mentioned the inhibitor. If you recall, for eukaryotes, the inhibitor is the alpha amanitin. Primarily it will inhibit the RNA polymerase 2, but it also inhibits the other polymerase. The strongest inhibition is for RNA polymerase 2, that is the alpha amanitin, which is obtained from a type of mushroom. You remember, the Amanita phalloides is the mushroom from where we get this substance, the alpha amanitin. Alright, so this is the inhibition of transcription both in prokaryotes and eukaryotes. This is a common question which is asked for 2 marks. Let us quickly go through the transcription once again, and then we move on to the translation. We said the transcription is divided into 3 stages: the initiation, elongation and termination. In prokaryotes, it is carried out by a single type of RNA polymerase which has 5 subunits: 2 alpha, beta dash and omega. This RNA polymerase combines with the sigma subunit to form the RNA polymerase holoenzyme, which is capable of the transcription, while the sigma subunit will identify the promoter and the rest of the polymerase will carry out the transcription. So this holoenzyme will scan the DNA, look for the specific promoter, whereby the polymerase holoenzyme will attach with the DNA, and then it will cause the denaturation, forming the pre-initiation complex of transcription. Then the RNA segment is formed, after which the polymerase can start moving forward. This is the promoter clearance. This is the last step of initiation. Once the RNA polymerase starts moving forward, we enter the elongation phase. So polymerase will move forward, it will cause the denaturation of DNA, it will cause the adding of the ribonucleotides, and the RNA will keep becoming longer and longer and longer. This is the elongation phase. The process continues till one of the two conditions are fulfilled. The first one, which is also known as the Rho or ATP dependent termination, is initiated with the transcription of the Rho binding site. Like I said, the Rho binding site is transcribed. Once it happens, the Rho which will attach will cleave the RNA from the DNA. Alright, the Rho subunit will cleave the RNA from the DNA. The polymerase is attached to the RNA. So the RNA polymerase which is attached to the RNA will separate, and this is the termination. This is first condition. In second condition, what happens? There is transcription of the self complementary GC rich segment, and immediately it is followed by the polyuse segment. In this case, what is happening? Immediately there is separation of the RNA from DNA without the need of any factor or the ATP. Alright, and once the RNA will separate from the DNA, the RNA polymerase will also separate, resulting in the termination. At the same time, we said in the eukaryotes we don't have the sigma subunit. There we have the transcription factors which are regulating the process of transcription. They are numbered and they can have the various subtypes also. So they are numbered and they can have subtypes also. One of the most important regulatory factors is the transcription factor 2D, which is made up of the two subunits, the TBP and the TAF. TBP stands for the Tata box binding protein, and TAF are the multitude of associated factors, the Tata box binding protein associated factors. So you have to remember about the TF2D. Lastly, we said the inhibition of transcription as primarily occurs in the prokaryotes. So here what happens? The two mechanisms. One is the inhibitor will bind to the beta subunit of the RNA polymerase. Examples include the rifampicin, rifamycin, streptoviricin, streptoglycidin, and the heparin. Another mechanism is by preventing the polymerase to attach to the DNA. So the molecule will attach to the DNA template. Example is the dactinomycin or the actinomycin D. In eukaryotes, one example of inhibitor of RNA polymerase is the alpha amanitin obtained from the mushroom Amanita phyloides. So this is about the RNA synthesis, that is the transcription, and the very important point, that is inhibition of the transcription. Inhibition of the transcription, as you should know. These are some of the commonly asked questions in your exam.